Hi, I'm Josh, and in today's video we're going to be looking into microscopy. Now within this we're going to be looking at the different types of microscope, the different way of preparing specimens to study them through the microscopes, and then some just general keywords within this area. So yeah, let's get into this video. The first thing we're going to look at is the microscopes. So the first microscope we're going to be looking at is called a compound light microscope. Now this consists of two lenses. You've got your objective lens, which is placed near to the specimen, and you've got your eyepiece lens, which is, well, you view the specimen through, so you put your eye on it, as the name says. And then, so these two lenses, they work together. The objective lens, it magnifies the object, and then the eyepiece lens, it magnifies that image further, so you get a really nice magnified image. This configuration of lenses allows for much higher magnification and reduced chromatic aberration, and so that's much better than a simple light microscope because you can get more detail, and they are illuminated by light that's underneath the sample. So yeah, that's how a light microscope works. Let's move on to the next one. Right, the next type of microscope is the electron microscope. Now there's two different types. You've got your transmission electron microscope, which you can write down as T.E.M, so T.E.M, and then you've got your scanning electron microscope, which you can write down as S.E.M. So they're just like ways you can shorten down how much you have to write in your exams. You just write down that little bit, as long as you say the full word first, and then you put in brackets S.E.M or T.E.M, whichever one you're using. That's absolutely fine, you won't lose any marks. But yeah, so in the T.E.M, a beam of electrons is transmitted through the specimen, and it's focused to produce an image. So it's really similar to the light microscope before, but it's just using electrons, which obviously are much smaller than light waves. And that means that it's much better for resolving, and it has a much higher magnification than a light microscope. Transmission electron microscopes have the best resolving power. They have a resolving power of 0.5 nanometers. That's an important number to remember because in an exam you could get asked to compare, and they will ask you to fill in the numbers. So yeah, that's the transmission electron microscope. Moving on to the scanning electron microscope. You've got a beam of electrons is sent across the surface of the specimen, and then these electrons are reflected and collected. The resolving power of a scanning electron microscope is 3 to 10 nanometers. A 3D image is produced from this because they're reflected and then being picked up by a computer, and then the computer is just processing that into an image, and it's a really cool 3D image, as you can see in the background on the right hand side. It just looks like that, and that's really valuable for scientists nowadays, knowing what something looks like. Obviously with antigens and all that good stuff, you can figure out a lot and cure diseases. So yeah, let's move on to the next one. Right, on this slide we're going to be comparing light microscopes and electron microscopes. So the light microscope is inexpensive to buy and operate. It's also small and portable. It's really easy to prepare, so when you're preparing your sample it doesn't get any distortions, any artifacts, meaning you can get a really clear image that is precise and accurate to what it actually should look like. And then a vacuum is not required for a light microscope. Natural colour of the sample is seen unless you're using a dye, in which case it'll be the same colour as a dye. Then you can get up to 2,000 times bigger with the magnification of a light microscope, and the resolving power of a light microscope is 200 nanometers. Specimens in a light microscope can be dead or alive, so it doesn't really matter which you use. Moving on to the electron microscope, they are very expensive to buy and operate. They are large and need to be installed, so they take up a lot of space. There's a very complex way to prepare the specimens, which often leads to distortion and artifacts being created, which isn't good because it reduces the accuracy of what you're actually looking at. You could completely change it by messing around with it. And then a vacuum is needed for electron microscopes. Black and white images are produced, but you can use a computer to colour the image, and then you can get over 500,000 times magnification with an electron microscope. That's a lot bigger than a light microscope. You also have the best resolving power with these electron microscopes. You can get up to 0.5 nanometers with a transmission electron microscope and 3 to 10 nanometers with a scanning electron microscope. And in this, the specimens are dead. The final microscope you need to know about is called a laser scanning confocal microscope. Now in this one, there is a beam splitter, which is a dichroic mirror. It only reflects one wavelength of light from the laser and allows the other wavelengths to pass through it. The position of the two pinhole apertures means that the light waves from the laser follow the same path as the light waves radiated when the sample fluoresces. This means they both have the same focal plane and hence the name confocal. Right, let's move on to the next slide. On this slide, we're going to be looking into the different mounts and stains that are involved in the preparation of slides. So, going into the mounts first, you've got a dry mount. This is where you get a solid specimen and you can section it. That just means slicing it into thin little slices. And then you place these onto a slide, put a cover slip over the top of it, and then just put it under your microscope and look at it. Then you've got your wet mount. This is basically the same as a dry mount, but you put a liquid on it. You also have to put the cover slip over the specimen at an angle so you don't squirt all the liquid everywhere, because that would then just completely defeat the object. The next mount is squash slides. This is the same as a wet mount, but you actually apply pressure to the cover slip using lens tissue. Now that squashes down the specimen. You can also do this by getting two slides, putting the specimen in between it and then squashing the two slides. Obviously you have to be careful doing this because you are using glass and it could easily smash if you apply too much pressure. So the next mount is smear slides. This is where you get the edge of a slide and you use it to smear across the sample over another slide until you get an even coating all over the slide. This is used to do blood counts and stuff like that. The next thing we're going to look at is staining. Now there's differential staining. This is to distinguish between two different types of organism or organelles within a single organism. Now you would only use this if the two things are really hard to distinguish. Obviously if they looked completely different, you wouldn't need to do it because you would already be able to tell what they are. Gram stain technique. Crystal violet is first applied to a sample, then iodine. This iodine fixes the dye to the sample. The slide is then washed with alcohol. The gram-positive bacteria retain the dye and are stained blue. The gram-negative have a thinner cell wall and lose the stain. They are then stained with a counter stain and then they appear red. So that's how you can distinguish between gram-positive and gram-negative bacteria. These two bacteria are involved in antibiotics. So penicillin will only work on gram-positive but not on gram-negative bacteria. So you have to figure out which bacteria you're dealing with by doing this test. The next technique is acid-fast technique. This is where you use a lipid solvent to carry dye into the cells. The cells are then washed with an acid alcohol solution, and if there's myobacteria present, it'll appear red, and if there isn't, it will appear blue. So the final thing is the keywords. Now, artifacts, I mentioned these previously. These are objects or structures seen through a microscope that have been created during the processing of a specimen. The resolution of a microscope is the shortest distance between two objects that are still seen as separate objects. Magnification is how many times larger an image is than the actual size of the object being viewed. Now, I haven't done a full slide on the magnification equation because it is so simple. All it is magnification equals the size of the image divided by the actual size of the object. The only thing is just knowing your standard form of your micro, your milli, and all of those, which you should already know. If you don't know them, then do some research because you're going to need that stuff. But yeah, thanks for watching the video. If you did enjoy it, be sure to leave a like. Comment if you've got any questions and you need some help. I can help you out in the comment section, and subscribe if you want more biology, physics, and maybe even maths and chemistry videos in the future.